Hello friends, welcome back to Dr. Jaggi's Academy. In last lecture, I discussed what are the mechanisms involved in producing ischemic cell injury. So now in this lecture, I will discuss what are the morphological changes that will take place in the ischemic injured cell. So now, as you remember, I discussed there are two phases of cell injury, reversible cell injury and irreversible cell injury. So I will discuss both what happens in the reversibly injured cell and what happens in the irreversibly injured cell. So first, reversibly injured cell. What are the morphological changes? I just want to tell morphological mean what are the structural changes that you can see under the microscope. That is the meaning of a morphological. So first of all, I am discussing what are the ultrastructural changes. Ultrastructural changes mean when you see a cell under an electron microscope, so those kind of changes they are termed as ultrastructural. So what happens if you see a cell under an electron microscope? So now, I discussed this diagram in the previous video also, I am going to discuss today also. So this is the normal cell. So you can see all the normal organelles, endoplasmic reticulum, ribosomes, nucleus, lysosomes, mitochondria. I am just discussing these things because they are important in terms of the ischemic injury. Now, but if you see, so I am just saying this is normal cell. Now this is the irreversibly injured cell. So what is the major thing? You can see this size of the cell is increased. This number one, cell swelling is the first sign. So cell swelling is the first sign. Second thing, you can say cell swelling. Number two, swelling of the organelles also. Not only the cell organelles. If you see the size of endoplasmic reticulum, you can see there is a much more size. So this is the swelling of organelles. Third thing, see this is the swelling. Obviously, mitochondria is also swollen. Now detachment of ribosome. I discussed in the previous lecture also. Ribosomes are attached, but ribosomes are detached. Third thing. Fourth one, blebs. Blebs are the changes in the plasma membrane because the water has accumulated. So membrane is not intact. It's tend to bulge actually, bulging of the membrane. So this kind of the bulges that appear in the plasma membrane, they are called the blebs. So this is the another morphological change. Then clumping of chromatin. If you see this kind of a nucleus, there is no clumping, but these clumps can be easily seen under the microscope. So these are the morphological changes in the reversibly injured cell. So I am just writing, cell swelling is the first sign, swelling of the cell organelles, mitochondria, endoplasmic reticulum, detachment of ribosomes from the endoplasmic reticulum, and alteration of the plasma membrane in the form of blebbing and distortion of microvilli, nuclear alterations. So these are the morphological changes in the reversibly injured cells. Now, second thing, irreversibly injured cells. So what are those changes? So this is normal cell, and this is a irreversibly injured cell. So what are the changes? First of all, I just want to tell, very important thing, this membrane is intact, but here you can see rupturing of the cell membrane. This membrane is ruptured, very important. If you remember the last lecture, I told this thing, this is a very important, rupturing of the plasma membrane. Second thing, lysis, because not only this membrane, whatever the membranes are present inside the organelles, they are also ruptured. If you see, this is swollen as well as this is ruptured also. This is not intact, this is intact, but this is ruptured, rupturing of this one. Then rupturing of the lysosomes also, because lysosomes will be ruptured, they will release the enzymes, but you can see rupturing. Then abnormal deposition in the mitochondria. If you remember, calcium is excessively deposited in the mitochondria. So you can see these kind of the abnormal deposits in the mitochondria. Then nuclear changes. I will discuss nuclear changes in this next slide. So these nuclear changes can be seen, which is called as pycnosis, karyolysis or karyorexis. I will discuss in the, so these are the, I will discuss again what are those things. Damage to the cell membrane, damage of the membrane of the cell organelles, ruptured lysosomes, abnormal deposits in the mitochondria, nuclear changes, which are karyolysis, karyopycnosis and karyorexis. So now I told there are nuclear changes. What are those nuclear changes? I am going to discuss those one. So first of all, karyolysis. What is a karyolysis? If you see a term, lysis, breakdown, karyo is related to chromatin actually. So let's say this is a normal nucleus. if the karyolysis takes place, what kind of a nucleus will appear? Now you can see this is the thing which is happening. So nucleus is ruptured, and you can see the staining. It is darkly stained, but it is lightly stained. Staining is reduced because the DNA is ruptured. It is damaged. So lightly stained with the basic dyes. You remember the DNA is stained with the help of a basic dyes. So when you add a basic dye, it is not stained. So you can see because it is not stained, so we are using a word fading because some color has been faded. Fading of basophilia of chromatin. Chromatin is the word we use for the chromosome chromatin, obviously, and we say basophilia because it is having an affinity for the basic dye. So we use the word basophilia. Fading of the basophilia of the chromatin is a characteristic feature of a karyolysis. So why this kind of a fading is taking place? Because this activation of DNase enzyme which destroys, which dissolves the DNA. So there is a degradation of DNA. So there is a light staining and fading of basophilia chromatin. These are the features of karyolysis. Now second thing, pycnosis. What is a pycnosis? Let's say again this is normal nucleus. If the pycnosis is taking place, now this is the thing which is happening. Now you can see the size of the nucleus is reduced and staining is increased. So you can say there is a shrinkage, size is reduced, but there is a staining is increased, darkly stained with the basic dye. So in the previous one there was fading, but in this condition there is increase. DNA nucleus is shrinkage of the nucleus. DNA is condensed into the solid shrunken mass, and there is a dark staining with the basic dye. And we say in the previous I said fading, but in this condition increase in basophilia of the chromatin. This is a feature of pycnosis. And remember, pycnosis is typically found in the apoptosis. I will discuss in the next lecture what is a apoptosis. But remember, in apoptosis there is a pycnosis. What is a karyorexis? Third type of nuclear changes. Karyorexis, let's say this is a normal nucleus. First pycnosis will take place, that means this kind of a pycnosis. Now that means pycnotic nucleus will undergo karyorexis, this kind of a thing. That means this kind of the nucleus is broken down into smaller fragments, fragmented nucleus. So this is the nucleus undergo pycnosis followed by fragmentation of the nucleus, and this is called as karyorexis. So this is what I want to discuss today in this lecture. So in the upcoming video I will tell what are the kind of the cell death. As I told, I will discuss the necrosis, I will discuss the apoptosis. Thank you.